Our next speaker is Dr. Mike Hogarty. Dr. Hogarty is an oncologist at the Children's Hospital of Philadelphia and professor of pediatrics at the University of Pennsylvania Perelman School of Medicine. He participates in international collaborations to improve neuroblastoma care, including leadership roles within the Children's Oncology Group, the COG, advances in Neuroblastoma Research Association, and the International Neuroblastoma Risk Group Working Group. His research laboratory studies how neuroblastoma blocks cell stress heading to the treatment resistance and what special needs these tumor cells have that might be used to develop new treatments. Dr. Mike Hogarty.
Thank you, Pat, and thank you for that nice introduction. And it's nice to see all of you and spend some time talking about a drug called DFMO and whether it's a drug that might be developed to be of use in treating neuroblastoma, what we know about it, what we don't know about it. If I am covering it at the level that is good for you, fine. If you want more detail, please interrupt. If you want less detail, please interrupt. Okay, so just something that you're all aware of, we don't do well enough treating our children with high-risk neuroblastoma. On the top of this slide is just a schematic of the current modern era treatment of lots of cycles of chemotherapy, followed by 1 or 2 transplants and radiation and bioimmunotherapy. And overall, if you're an optimistic person, cup half full, you would look at those survival curves from the 1980s onward and say, you know, they're making progress. It's only recently that the 3-year survival for high-risk disease has crossed the 50-50 mark. So that's kind of terrible. I'm an optimistic person in general, but in pediatric oncology, I tend to see the other part of the cup, which is how many children we're failing to cure with our current treatments. And even the ones that are getting cured pay a price in terms of toxicity. So we really do need new approaches, and we're developing them. This isn't a new idea. The whole idea of using an antibody against tumor cells, the dinutuximab antibody, has been developed over the last 15 years, and it's very successful. So there's a long history of finding new ways of going after the target, which is the tumor cells. So one of the new ways at which we can start to ask what are good targets is using the power of the genetics or genomic technologies to look into the tumor cells and say, what mistakes do you have? So at one point, all neuroblastomas were actually a neural crest cell that wasn't malignant, but they had mistakes, mutations in it, and those mutations are gathered up until they're sufficient to drive a cell into growing into a tumor. Sometimes that also leads to a tumor capable of metastasizing or spreading. And we can take tumors from different times and do sequencing of the genome and begin to say, what is it that's driving you? That's a big term in sort of precision medicine. What's the driver of the tumor, and can we target it? So when you do that for neuroblastoma, you begin to sort of create a list of genes that can be found. And among them are the MYC genes. MYCN, the N means neuroblastoma because it was discovered in that tumor. Its very closely related gene is just called MYC. And then there are some others. Yael just spoke about ALK and her work targeting that kinase. But basically, these are your driver genes. And then there are some genes that aren't typically mutated at the beginning but might be acquired. And I think Yael's also mentioned the experience with P53. So that the current approach is all well and good when it works, but there are problems. So the idea would be find out what the driver is for a tumor, pick the drug and block it. But what about genes like telomerase, ATRX, some of these chromatin remodeling genes, genes that you don't really have to know anything about other than people who try to drug them say they're not really druggable. We don't have a drug for them yet. What do you do then? Also, it's hard to develop a drug that works. if you take neuroblastoma that's already a rare tumor and say, well, now we're going to break it up into 25 even rarer subtypes because we have to develop a solution for each specific genetic problem, it gets to be daunting. But what we're going to talk about a little bit today in the context of DFMO is that gene called ODC1, or ornithine decarboxylase. We'll come back to that. The reason that we sort of circle MYC and MYCN here is it's really one of the most common drivers. So there is a problem of some of these genes being in small subsets, but at least 40% of tumors that are high risk have MYC amplified, and it's one of the tests that every country in the world does, essentially, to try to characterize the tumor for at least that one gene test. Even if you didn't do sequencing, almost everybody knows that, oh my gosh, we were diagnosed with neuroblastoma, and the doctor said they're sending a gene test that might make a big difference in whether they consider it a bad tumor or a good tumor, as if there is any good tumor. But MYC is a particularly aggressive driving gene. So if you take all high-risk neuroblastoma, like on the last slide, MYCN amplification occurs in about 40% of them. What I mean by MYCN amplification is all of the genes in our cells should come in 2 copies, and only 2. You got one from dad, you got one from mom. And that's true of every chromosome and every gene. They should be 2 and only 2. Here's a normal cell. And you're actually labeling the MYCN gene with a little probe that turns green, and then a control gene that isn't MYCN, but it's on the same chromosome, and every normal cell looks like that. Mom and dad's MYC copy of the gene is there in 1 copy each, and the normal dosage is there. And this is what it looks like when you do that same test on an NMYC amplified tumor cell. That's not subtle. We're not talking about a 3rd copy of this bad gene or a 4th copy. We're talking about literally dozens and hundreds of copies of this very aggressive tumor gene. And so it can be quite striking. The other gene that's like MYCN, MYC, can sometimes be amplified, but not nearly so much. I'm showing ALK here. I'm showing some of the other mutations. But this big outer circle, you can see, has what I call MYC signal high. Okay, because I'm getting in the way of your video. Gotcha. I'm going to stay over here and use my laser pointer. So the MYC signal high, I mean, that's basically, I put every tumor in there, and that's because there's lots of ways to get your MYC turned on in a tumor cell. The amplification way is so apparent to us, it was hard to miss. And that was in 1983 we discovered that. But we're slowly adding to other ways at which the tumor cells are turning on the MYC in a way that sort of drives them. So MYC is the most important cancer gene, I would argue. It's very commonly turned on, aggressively correlates with aggressive tumor behavior. And to prove how important it is in making neuroblastomas, if I just take the NMYC gene and I express it in a fly, a fruit fly, in their nervous system, I make neuroblastomas. If I express it in a fish in their nervous system, I make neuroblastomas. If I express it in a mouse in their nervous system, I make neuroblastomas. That's kind of the proof positive that that is a driver gene, because we're not doing anything else other than giving it MYC. Unfortunately, we haven't yet come up with a way to turn it off. So since 1983, we've known it was important, and in those 30-plus years, we don't have a drug. It would be one of those categories, the question earlier, what if you find a genetic mistake that you can't drug? We do that every day. We find kids with NMYC amplified tumors that we can't drug. So the question is, are there other ways at which you can go after this biology with treatments? And so why MYC? What's so great about MYC if you wanted to make a cancer? Well, MYC does something incredibly important in biology. It coordinates the decision for a cell to become 2 cells. So it's a transcription factor. What I mean by that is it stays in the nucleus where all the genes live, and it turns them on selectively. It doesn't turn everything on. It turns on a whole bunch of different genes all at one fell swoop. And what that does for a normal cell is it helps really tightly control that very important decision of when should I grow and make 2 cells and when should I just sit here. So it does that because it turns on all the genes that make the proliferation or the division cycle happen. And then that's very costly. You got to make a lot of stuff because you don't want, if you're a tumor cell, you have to make 2 cells of the same size as the one you started with. So you have to build all those building blocks, proteins and everything, and you need a lot of energy to do it. And cancers hijack that. That's what's relatively efficient, and why MYC is so commonly selected as a cancer driver is because if you're a cell that turns on MYC, you get all of that in one fell swoop. You drive your ability to become 2 cells, and you drive all the machinery that helps you make the mass and to help you get the energy needed to do that. And MYC's doing that by turning on literally thousands of genes, and I'm showing you below all the different things it's controlling. That's why it's sort of a one-stop shopping for an aggressive growth behavior. And I'm going to talk today about polyamines and proteins because that's a really important part of what MYC needs. Between half and 3/4 of every cell of our body is protein. So to make 2 cells equal to the parent cell, you've got to build a lot of protein. And polyamines are critical to doing that. And if you're wondering what polyamines are, I'm going to get there. We stumbled upon polyamines many years ago, and I had no idea what they were. So after we started thinking about it, we did a lot of homework. So I'm going to sort of condense that and give you a quick primer on polyamines shortly. But I want you to have the idea that MYC has to drive all these proteins and that they need polyamines to do that, because that's our possible sort of point that we can exploit. Okay, you can take tumors and you can not only look at their genes for mistakes, you can also read out the genes that are turned on and which ones are turned on high. And there's a variety of sort of boring-looking, colorful pictures that people put in their articles all the time that are showing how that's working out. But I just want, the point here is that there are lots and lots of studies of different cancers, including neuroblastomas and even normal cells, where you're turning on the NMYC or you're turning on the other MYC, and time after time after time, the signal that's jumping out, and I already alluded to it, is the protein machinery. And in our own work with neuroblastoma, the reason we became polyamine interested is because we kept seeing genes like ornithine decarboxylase, or ODC1, and all of the other genes that regulate polyamines. And so we wondered if they might be important in neuroblastoma because we'd never really thought of polyamines before. And the literature from 20 years before had already taught us a lot. First, ODC1 is turned on by MYC. So that's been known since the 90s. In fact, when I started in the lab, when we wanted to prove whether the tumor cell really was using MYC or not, we would look for how much ODC it had made. We didn't consider it an important thing. It was just kind of a marker. ODC, though, also can turn a non-malignant cell cancerous, which is another strong sign that it's doing something bad in terms of driving the behavior towards tumors. And it's basically the drug target of DFMO. So MYC is commonly turned on, which means ODC1 is commonly turned on, which means maybe that's our inroad, because if we can't drug MYC, maybe there's a way to drug this enzyme. So what are the polyamines? The chemical names, they're putrescine and spermidine and spermine. Basically, they're kind of chaperones. They're really important for how DNA and RNA can stay stable. And without them, you really have a struggle to be a good cell and certainly a struggle to proliferate. So if you measure polyamines in any tissues in our body, the tissues that are growing have lots of polyamines and the tissues that aren't have very little. And that's mirrored exactly in cancers. So rapidly growing cancers have lots of polyamines. The more slowly growing ones have less polyamines. And the polyamines are really important for how proteins are made in tumor cells. You know, coming back to that, MYC is trying to drive you to make a lot of proteins to help make more tumor cells. And people have been studying the polyamines in the cancer context for a long time. So it was new to me when we saw it in our experiments, but it wasn't new to scientists. They had been thinking about how can we do something about this biology. So this is a flickering, is it flickering for you guys? Yeah, so it's a flickering version of polyamine biology. So ornithine in blue there, that's just an amino acid. That's a building block of proteins. And ornithine decarboxylase, the ODC gene, takes ornithine, snaps a piece off of it, and makes putrescine as a polyamine. And I will stop reaching and use that. And then these other enzymes in green turn the putrescine into some of these other polyamines. And the red enzymes are kind of blockers. They go in the other direction. So the green things in those boxes are genes that are helping a cell make more polyamines, and the red things are the counterbalance genes that are trying to get rid of polyamines. And so we've looked in neuroblastomas, we've looked in about 600 plus neuroblastomas and the genes they've turned on. And this is just showing you that the survival over time, so you've looked at survival curves, I imagine. So we wish they all stayed up here, which is everybody stays alive. But unfortunately, with high-risk neuroblastoma, children relapse and die from this disease. All we've done is we've taken 650 patients and split them into the half that has the high ODC1 in this case, and the half that has the low ODC1. And you can see how much worse the curve is for the high ODC1 group. And then we did that for the next polyamine gene and the next, and you see a pattern repeating itself. And then when we do that, looking at the genes that are trying to take polyamines away, it flips. So the bad tumors now have the low ones. They're low expressing because these are the genes that would deplete polyamines. So it really looks like these tumor cells are going out of their way to create polyamines. And it matters because your prognosis is matching with what your tumor is doing for polyamines. And to say it a different way, if I give you a score for just how polyamine driving your tumor is and broke you into 4 different groups from the highest polyamine making to the lowest, it mirrors your survival outcome pretty impressively. And that means that kids with MYC driving ODC and all these other changes are going to be in these groups, and that is scary and yucky, but at least we have a drug that might work. The other thing about ODC1 is it's not just that MYC turns all these genes on, it's that, going back to that very first slide, that gene itself is also amplified in a subset of tumors. So in a scan like this is just reading across all the genes in an area of DNA. And when it spikes up, it means there's lots and lots of extra copies. And so here's a classic MYCN amplification in this kind of technology. But ODC1 is equally amplified in this tumor. And about 6% of high-risk neuroblastomas not only have turned on NMYC by amplifying it, they've turned on this ODC1 gene. And I'm just showing that in an updated version of what we looked at earlier. So a lot of these high-risk tumors are driving polyamines in lots of different ways as if it means something to them. So this is a quick summary so far. The MYC genes are really important, but we can't drug them. We think what they're really trying to do is coordinate growth with all the things you need as building blocks to make a new cell. The polyamines are intentionally and quite coordinately regulated by MYC to help do that. And the more turned on a tumor cell's polyamine-making system is, the worse it's going to behave. And nature spent a lot of time investing in this pathway. You notice there's lots of different genes regulating polyamines, and because of time, I didn't go into all the checks and balances where one gene's turning an enzyme off, but there's another to turn it down and another to turn that one down. So there's a reason that this is very tightly regulated. But what does any of this have to do with DFMO? Well, as I said earlier, DFMO is a drug that targets ODC1. So DFMO stands for difluoromethylornithine. So this is ornithine, and if you put this little methyl 2Fs group, floral groups, stick it on right there, you've got DFMO. The enzyme is looking for this. Remember, it's going to break a piece off and make putrescine. But if you give enough DFMO, it's going to find this first. And when it tries to break this piece off, it sticks. And it sticks forever. DFMO will never let go of ornithine decarboxylase, so it can never make this polyamine step. So it's what we call an irreversible inhibitor, which is a good thing for a drug because you don't have to worry about whether it stays on long enough. Once it finds its target, it's there for good. And we know it has anti-cancer value, at least in certain contexts, because if you're an individual that might be at risk for colon polyps or adenomas, those are precancerous, and if you have one of those removed and it's not cancer, you're not going to get cancer treatment, but your doctor is going to tell you, we got to keep an eye on you because you have a high probability of getting more of these, and they could become cancerous. And if you use DFMO and another anti-inflammatory medication together versus placebo in a blinded study, the DFMO-treated patients have a marked reduction in having an adenoma come back and a really, really high decrease in ever having one of the more bad, aggressive cancer-like adenomas. So we know in certain contexts of prevention, it can be quite useful. So we're, though, asking a slightly different question. We're not trying to be great to prevent, but can it do anything when you already have a tumor that's using polyamines? There's a mouse model of neuroblastoma where that same human NMYC gene, or MYCN, is turned on in the nervous system. I alluded to it earlier. And those mice all get tumors. And they get tumors that look all the world like a neuroblastoma in a child under the microscope, by their genetic mistakes, in many, many ways. And we've shown that the way they turn polyamines on also mirrors what we found in the human tumors. So we thought it was a good model to ask the question about, what's MYC doing? How much does it need ODC1 to do it? And can we block it and get some evidence that maybe it's a useful drug? And so the first thing we did is we said, well, let's block it before they even have a tumor. Let's do something that's not really clinically relevant yet, but might be very important to get a sense of whether it's even on the right path. And that is to put some DFMO in the drinking water of these mice when they're born. And they're 100% destined to get tumors, but are they if they're drinking DFMO along the way? And the sad answer is that, yeah, they're still going to get tumors, but they live 50% longer. And all we did was add a modified amino acid to their drinking water. We didn't give them shots. We didn't give them anything. They grew well. They were fine. They just had a special water. And their lifespan was doubled because the tumors were growing less well. So we thought that was kind of cool. And then we added a second inhibitor. This one's not really a drug, but you can give it to mice, and it blocks one of those other green enzymes, the one called AMD1. And now when you give both of those together from birth, half the mice don't ever get a tumor. And all those drugs are doing is blocking polyamines. So that's, to me, a pretty profound signal. Again, it's not anything you carry across the street to the clinic, because it's a different system, but it tells you that maybe there's something here. And then the next thing we did is the more relevant question. We said, okay, let's let the mice get their tumors. They live for, by the time you can feel the tumor in their belly, they've got about 7 days to live, because these are very rapidly growing tumors. And then we just threw some DFMO in their drinking water, and they always live longer. If we combined it with chemotherapy, just to try to give them a little more time to survive how aggressive the tumor is and see if the DFMO could do something, we saw things like this. Is this dying on me? This is the DFMO chemo arm. They always did better, sometimes strikingly so. And when we stop treatment, they don't get the tumors back. So these are mice that had a tumor, typically should have only lived for another 7 days, got chemo that wasn't going to cure them, or maybe would rarely cure them. But when you just added DFMO, they lived longer and often were what we would say in the world of mouse cancer cured. Okay? And one of the things that we always have to remember is we don't just want to cherry pick one or two models and then say, oh, we've solved the problem. We have this mouse that we can fix. Because that leads to not really being critical enough and not looking deep enough. And that's been pointed out in numerous articles and editorials about how our preclinical models don't predict so well. And some of the take-home messages are, don't just use one model, use complementary models. Look really, really hard, really, really deep, really, really broadly. And so we did that. You don't have to read any of this, but each little line here is a different mouse experiment. And if it has a blue asterisk near it, it means the DFMO improved the survival in that model. So it's not 100%, but it's striking how often it improves survival. And that involves both the mouse models and then also models where you take human tumors and grow them as xenografts. And different kinds of human tumors. It's not just MYCN amplified tumors either. So what don't we know about DFMO? This all looks pretty good. Why isn't everybody getting DFMO? Well, there's a lot of things we don't know. Who should get it? Should everybody with a neuroblastoma get it? Or is it only going to work in the kids where the MYCN's turned on really high? Or maybe the ODC has to be amplified. Maybe that little 6% is where it's going to work great. We have to learn more about who should we expose to DFMO. That's a smaller problem. That's a solvable problem. That's what clinical trials should answer for us. The harder things are things like what dose should we do in those clinical trials? The low doses are very appealing because they're very non-toxic. The higher doses are appealing to me because I'll show you some of the model information that tells me that's the dose that actually would be effective. But I can't say we really know for sure. When should we use it? Should we combine it with chemotherapy from the get-go? Should we wait till the end of your treatment when there's not going to be that many tumor cells left and we're just trying to mop up with something that's not so toxic? Should we combine it with other drugs that might make it work even better? We don't know any of that. But we do know that we can begin to chip away by using our available experimental models, but in the back of our mind, mice are not little people. They're not children. The models are just models, and at some point, you have to take it into the clinic and see how it works with real children with real diseases. And we owe it to families to do as much of the homework as possible. That balance between you can't drag your feet forever, we need things now, versus wouldn't it be a shame to rush into the clinic with the wrong dose or the wrong strategy, because people aren't going to be patient forever. And if you have a couple of non-effective trials with DFMO, then DFMO is done, right? And it's true of any drug. People are going to move on. So we really need to get it right. And one of the ways we can try to get it right is just really understand what it's doing. I mean, so far we've really been talking in that general level of it's bringing down polyamines so you can't make proteins. But we know how it interferes with proteins. So just a quick little example, not to bog down in basic sciencey stuff, but there's this gene called EIF5A that requires polyamines to be turned on. It gets this thing we called hypusine attached to it, and that's from a polyamine. So if you have no polyamines, this gene is completely off. And if you do have polyamines, it's on, and it's one of the most important proteins that helps you make other proteins, okay? So we can actually do different doses of DFMO, and if this is really important in why the drug works in the animal models, then you and I would all look at this and say, I sure don't want to be dosing at 150 micromolar in the blood. I sure want to be at least at like 250, 300, right? So this is just an example of how you can use this kind of data. Similarly, we know that polyamines help the immune system be either suppressed or active. And it does so pretty profoundly. So that antibody GD2 doesn't kill neuroblastoma cells. Even though the trials show it improves survival, all it can do is stick to neuroblastoma cells. The reason it improves survival is because the NK cells and cells like them can now see the tumor and kill it. And when we give DFMO, NK cells go way up. And we can also then say, okay, well, what dose in the mouse doesn't rise the NK cells and what dose does? And you can begin to sort of explore what you think is the most important doses to bring into the clinic. And you can also just look at the effectiveness. So this is one of those trials with the MYC mouse where we're using different amounts of DFMO in their drinking water. We can measure what that leads to in their bloodstream, so we know what they're being exposed to. And I can show you that the only time you live longer in this very simple experiment is with the high-dose DFMO, when you get your levels up over 150 micromolar. And that's about 6 to 7 grams per body size per day. And everything else doesn't do that. You don't get any benefit. And so when you start looking at the human trial data from consortiums like NMTRC, Beat Childhood, I think they're called Beat Childhood Cancer officially now, but a lot of people still remember this term. NANT is another consortium that does sort of trials, phase 1, phase 2 neuroblastoma trials. And there are a number now of DFMO trials. And when you use it at lower doses, you get predictably lower levels in the bloodstream. And then when you use it as higher doses, you get higher doses in the bloodstream. So this doesn't answer the question about what dose is going to work. We don't know what dose is going to work, but it allows you then to say, well, if you want to turn the protein off, or if you want to make the NK cells up, or if any other thing you think is really critical to help DFMO kill a tumor cell is happening, you can at least say, well, if I need a certain concentration, then this is the dose that we have to be giving the kids. Okay? So where are we going with this, thinking about it? So I was involved with the NANT trial, the one at the bottom with higher doses, and the reason is because I have worked in those models that tell me I think we need higher doses. I love the idea of giving non-toxic doses, but if they're not going to work, then non-toxic doesn't get us anything. So we intentionally designed it to be a chemo protocol with high doses of DFMO. And there were responses. It was tolerable. We know the doses can be combined with chemo. And so now that's being handed off to COG, which is a larger consortium, more hospitals, more children across the country and Canada and elsewhere access to a COG trial. And it'll be a randomized phase 2 study where they get what was the winning arm of the last really important phase 2 trial that many of you probably have heard of, where children who had relapsed were given irinotecan and temozolomide chemotherapy, and then they might have gotten antibody, dinutuximab, added to it. And it had a remarkable response rate. Well, that is the regimen that's now the backbone of this new trial that would hopefully open later this year, and you will be randomized 50-50 to get DFMO or not get DFMO at the high dose. And that's a design we like for 2 reasons. One is, at the high dose, we know DFMO is antagonistic to neuroblastoma growth for all the reasons we alluded to by way of proteins and what MYC is trying to do. But also, if it has the same effects in children's tumors that it has in mouse tumors, it might change the immune cells in the tumor in a very favorable way. One of the reasons we think immunotherapy doesn't work all the time is because the antibody can find and stick to the tumor cells, but your immune system is just too weak or ineffectual at doing anything about it. And tumors are smart. They can create chemicals. They can do things that turn the immune system down. And if DFMO does a lot of the things that we believe it does and can prove in the lab it does, then it's going to change some of those dynamics and hopefully goose the dinutuximab too. So it can work with the chemo, it can work with the antibody, but all of that is conjecture, and only the trial in a randomized design is going to say, well, did it do any of that? And that's kind of an example of the way we've been thinking about DFMO and bringing that drug forward. You know, a lot of preclinical work using complementary models, try to get a sense of what doses do you need, how do you combine it, trying to find out predictive things, what subset of tumors might it work best on, is there a test that you can do to tell me that my child should get it or shouldn't get it. Those are all things we're still working on. Go to the clinic and sponge up as much information and learning from the actual children who took the drug as part of a trial. Get that information, feed it back into your models, and that cycle is how we develop more thoughtful and hopefully more effective treatments over time. And so I'll just leave this slide up. This is a lot of people that have helped along the way and the funding agencies that have been so important to sort of supporting this kind of lab work. And I am very happy to take any of the questions you might have. Thank you for your attention.
Does anyone have any questions? Okay.
Hi, my name is Andrea, and I'm from Minnesota. And you mentioned polyamine signature scores, and I'm wondering if that is some sort of analysis that is done in most hospitals. No, it's not something that's readily available, or can you speak more to that?
Sure. It could be done. It's nothing more than a measurement of the expression of a handful of genes that we sort of picked because we knew that they regulated polyamines. If you look at that, that would tell you that high scores are bad, low scores are good. They do tend to track with other things that we already know. So obviously when your MYC gene is amplified, because it's regulating all of that pathway, they tend to be really high already. So we kind of know if you're MYCN amplified, how that test would look on you. It's possible that there are kids that we miss, that we think, oh, you're not high risk. But had we done that, we would say, ooh, your polyamines are really high for some reason. So it's probably something that deserves a little further thought.
And then the second part of that question is, as a parent of a child who is not amplified, we've completed vaccine and gone on to DFMO, but this data makes me question whether or not DFMO is relevant for a child who is not amplified.
I think all parents are always struggling with, you know, is it the right choice? Are we doing the right things? That's in human nature. It reflects our deficiencies as the team of people trying to guide decision makings and providing good treatments. I would say that in our models, we have plenty of tumors that don't have NMYC amplification that respond to DFMO beautifully. That probably speaks to the fact that NMYC amplification is only one very obvious way at which a tumor cell turns on MYC. But you know, if you go back to that big circle chart, everything had MYC on. So I'm a MYC person, so maybe I'm a little MYC-centric in how I look at neuroblastoma. But there's lots of different, we know this now with lots of fancier genetic tests. You don't have to turn on 200 copies of the gene. You could break 2 chromosomes and put them together, and that just turns the MYC way up. You can turn the C-MYC up, you can turn the N-MYC up. There's lots of ways. So I would have no reason to tell you that a non-amplified tumor can't benefit. So, and as far as dose, we don't know whether the higher doses are going to be better. So I think that trial design for any one child is perfectly fine. If it has flaws, I think some of it has to do with, well, what are we going to learn from it if you're not randomizing it, right? What are you going to compare it to convince the world it worked? That gets to be a little bit trickier. But those are all things we're all, as a community, struggling with to try to get that information.
Hi, my name is Helen. I'm also from Minnesota. Going back to your circle graph, my child is HERX, and I saw that HERX was kind of on your circle. Mm-hmm. How does that play into all of this process that you're describing?
Well, so the one thing we know about ATRX mutated tumors is that they don't have NMYC amplification.
Right.
They're mutually exclusive ways towards getting at that tumor type. But they do have, if you look at the output of MYC, if you measure the genes that MYC would turn on, it still has a MYC signal. So again, I think we're still in early days, and I would not at this point be willing to exclude any genetic type of tumor from thinking about whether or not these drugs could work.
Okay, thank you.
Yep, sure.
Okay, one more question? Oh, okay.
Sorry, is that okay? My name's Laura. I'm from Canada. Our daughter just started the DFMO trial in Michigan, and there was a lot of kind of back and forth about the low polyamine diet that goes along with it. And I guess I just wanted to see what your thoughts are on how much that would affect taking the DFMO and how it would react.
So the theory behind that is that there's lots of polyamines in our body. I mean, we've already reviewed that all cells that are growing are making a lot of them. And there's polyamines in dietary substances, some foods more than others. And if you follow a polyamine restricted diet, you can measurably decrease the amount of polyamines in your blood. But you don't go from high levels to nothing. You just go down measurably, but we don't know whether that's meaningful. We also don't know whether it changes the important part, which is in the tumors. Are the tumors still polyamine rich because that's where the MYC is turned on and that's where they're intentionally making them? Or are you just going to make a child miserable by telling them, look, you've been through a lot and now we're going to make you taste this stuff and restrict things that you might like? So if you looked at your child's diet and it polyamine poor just by their preference, I'd say, great. If you look at the diet and you say, I want to get rid of this, this, and this because it's, quote unquote, high in polyamines, I wouldn't necessarily be all that keen on it. I wouldn't say you couldn't, just I wouldn't have a reason to think it was going to be a really important factor.
Thank you.
Yeah, sure.
Do one more question then? Did you want? Okay.
So my question was about the NK cell. I was wondering if you actually measured that data yourself as a prior work that was done before, and all the data on the mouse wouldn't be relevant to any impact on the NK cells in humans, and whether the lower dose has an impact on the NK cell or not.
Yeah, so the last question first, it doesn't. So the lower dose doesn't change the NK cells. We did that work ourselves. What we do is we take the tumors out of the mouse at different times. We can sacrifice them early when the tumors are small. We can sacrifice them at the very end when they're dying of their tumor. We can make a suspension of all the cells, and then we can sort of put them in a machine that basically characterizes and counts each one, T cells, different types of immune cells. What I didn't tell you, because I was trying to keep it sort of general, is that not only are the NK cells increased in their number within the tumors, but they have different functions that you can infer based on what's on their surface. And NK cells can turn on receptors that make them more active. They can also have receptors that make them inactive. So DFMO does two things. It increases their number in this model, and it helps them put on more of the active I'm functional receptor, and then the tumor cells are also expressing more of the come find me NK ligand, the things that the receptor would want to seek out and bind. So is that the reason it works? I don't know. Maybe that's all set and ready to kill, but something's not letting the NK cells kill. That sounds kind of sad because it means that it's not working, but to me I'd say, well, that's another opportunity. If we already know it's improving your life and making tumors smaller, what if we can solve that riddle? What if we can take these NK cells that are all ready to kill but for some reason not, and find out why they're not and fix that step? So I think there's lots of things we still have to learn to get it just right as we go. Yeah.
All right. Thank you,
Thank you.
Dr. Hogarty.